Hi, everybody. My name is Mara Carroll. I'm the Development Officer here at Jet Foundation. Welcome to Jet Foundation's Community Webinar Series. We're excited to have our friends from Edgewise Therapeutics here today joining us for our webinar. We are going to wait just another minute or two till we have a few more people logging on. So hold tight and we'll get started in just a minute. Hi again, everyone. This is Mara at Jet Foundation. Welcome to our community webinar series. We are going to get started in just another minute or two. I see a lot more people logging on to join us today. So maybe one more minute and we will open it up and begin our webinar with Edgewise Therapeutics. So just another minute. Okay, so hi everyone. My name's Mara Carroll. I'm the Development Officer here at Jet Foundation. Welcome to our community webinar series. We're so glad you're all here joining us today. We're excited to have Edgewise Therapeutics here with us today to introduce their company, their science, their program in Becker and Duchenne. So looking forward to their webinar in just a minute. For those of you who haven't joined us before, our webinar today will be about 30-35 minutes of the presentation, and then we will have questions afterwards. So there is a question box on your screen. Feel free to ask questions throughout, but we will get to them all at the end of our webinar. So with that being said, again, thank you all for being here, and I will introduce now Abby Bronson, who is the Vice President of Patient Advocacy at Edgewise Therapeutics. There you go.
Hi, everybody. Welcome. And just first of all, I really want to thank the Jet Foundation for setting us up and giving us the opportunity to communicate with all of you. So thanks very much. The other two speakers, I'm just going to introduce our other speakers. We have Alan Russell, who is our Chief Scientific Officer. He also was a co-founder of Edgewise, and you're going to hear from him here a little bit later. And also Joanne Donovan, who is our Chief Medical Officer, and you will hear from her as well. And many of you know Joanne from her previous life at Advocacy, and we are so lucky to have her. So this is our forward-looking statement. I am not going to read the whole thing. I think probably the most important thing is that our compound is an investigational drug and is not approved in any territory. So just a little bit about Edgewise as a company. We launched in 2017. We're based in Boulder, Colorado, as you probably know, and Alan, who you'll hear from, was one of our co-founders. Our sole purpose is to improve the lives of patients and families who are suffering from rare muscle disorders. We have an approach that is very unique. I think you'll hopefully understand that by the end of our presentation. And it's potentially complementary with the current approaches, and it directly targets key muscle modulators. Our lead program, which you'll hear it being referred to as EDGE 5506, it doesn't have a name yet, it's focused on Becker and Duchenne muscular dystrophies. We've worked hard since 2017, and we have successfully initiated our phase 1 study, so we're really happy about that. And before we get into the compound and our program, I just thought I'd take a few minutes to talk about patient advocacy at Edgewise. And the whole premise is that we can do our jobs better when we listen and we learn from you. So that's basically what I try to do at Edgewise. And how do we do that? And what do I mean by that? How does your input really impact what we do? So we do a lot of focus groups and surveys. Maybe many of you have participated in some of those. And we gather all that input. And when we listen to you, we can make our clinical trials designed so that they're more appealing, so we can enroll faster, so we have fewer changes that we have to make to the trial as we go along. Those are called amendments. So that input that you give us is really, really important, and we try to listen to you. There's also something called the therapeutic context for risk-benefit decisions, and this is part of the patient-focused drug development effort at the FDA. So when they're making a decision, balancing risks and benefits, it's hard to do that without really understanding the burden of disease. What's the treatment landscape out there for that disease like? Is there something out there? So the input that you give us can really help establishing that therapeutic context as well. We also want to be a trusted partner in the Duchenne community, and so we work with all of the advocacy organizations, the Jet Foundation and all the other ones, both here and in Europe, around the globe, really, to align on how we can really help the community move forward and what our shared goals are. We're all in here to do basically the same thing. So if working together, we can get there faster. We can serve as a bridge with our connections and who we work with, connecting the research ecosystem and really pushing forward to help find better and new treatments. And also creating awareness for the disease and helping educate individuals and families is also part of what patient advocacy at Edgewise does. So I think at the end of this presentation, I have an email, and we really encourage you. Each one of you is important. And if you have a question or a comment, or just feel free to reach out and one of us will get back to you. So now I'm going to pass it to Alan, who will take you through a little bit about the science of Edge 5506.
Thank you, Abby.
You're welcome.
No problem at all. So we are not taking a genetic approach to Duchenne and Becker. We're taking a structural approach to try and reduce stress in skeletal muscle. And that unfortunately necessitates me giving you a little bit of background on what the true role of dystrophin is and how that's impactful if you have Duchenne or if you have Becker muscular dystrophy. So you've probably seen a version of this slide innumerable times. And it's my reminder just to give you an overview of where dystrophin sits and what it does. So that purple complex there in the bottom right-hand side, that is the dystrophin complex. The dystrophin protein is, of course, the long linker protein that connects all the internal machinery of muscle to the extracellular domain. So all the protein that surrounds muscle. So I'll just, rather than distracting you with the text on the left, I will read it just so you can get it too. So when dystrophin is deficient or absent in Becker and Duchenne muscular dystrophy, the stress of everyday use of muscles triggers muscle damage that ultimately leads to loss of function. So it's not the inherent property of the protein that causes weakness. It's the eventual breakdown of muscle and all the fibrosis. The myofibrils that make up muscle have important proteins called myosin, and the reason I'll talk about that, it becomes self-apparent in the near future. And the interaction of myosin with other proteins causes muscle contraction and delivers the power to muscles that conduct everyday activity. So I think most of us inherently understand that muscle's role is to contract and move us about. I'm just going to fill in a little bit of detail on some of the specifics behind that movement and why it's so impactful in Duchenne and Becker. Here's some graphical images taken at very high power of muscle, and it kind of illustrates how dystrophic muscle breaks down. When dystrophic muscle contracts, injury occurs. And you can see a normal organized muscle there on the left-hand side. And what happens visually to that muscle when it contracts? Contraction is really the starting point of all of the problems. And what happens if you don't have dystrophin is you get membrane stress. As I mentioned, dystrophin is this connector between the inside and the outside of the muscle. When muscle contracts, when it doesn't have dystrophin or the dystrophin is truncated inside, you get internal stress and you get disruption of all of the contractile stuff. That causes membrane stress, and then calcium starts entering the muscle. Now, calcium is required for all sorts of functions, but in the context of muscle, if you get too much, you get myofiber death, so the muscle fibers break down, and then you get hypercontraction as well. And you can see that occurring in those two pictures on the right-hand side. And that really triggers the breakdown of the muscle fibers. And the only way that the muscle can come back at that point is by regenerating a new fiber. It's amazing how good muscle is at doing that. But of course, over time in Duchenne and Becker, that breaks down, leading to the replacement of muscle with fibrotic tissue, things like fat and collagen. On the next slide, I'm actually going to show you a video of that occurring. This is on YouTube. So you can go to YouTube and just type in, I think it's a lumbrical contraction, which is technical, but what we're looking at here is dystrophic muscles contracting. They're so small, these are dystrophic muscles from a mouse, you can see right through them. You can see on the left-hand side is the normal, and on the right-hand side is the Duchenne equivalent of muscle. There's no dystrophin. And as that muscle is contracting, you can see that this stress is manifesting in the fibers getting chopped up, and then they're crushing in on themselves. This kind of hypercontraction that essentially leaves an empty tube that the muscle then has to grow a new tube into. Okay, so you can see it's pretty profound injury, and you can imagine with time that causing significant problems to overall functionality. Now, we are made of different types of skeletal muscle fibers. You may have heard this before, just in everyday talk of exercise and the like. Our skeletal muscles are made of both fast and slow skeletal muscle fibers. Okay, the reason that we have different types of fiber is it allows us to coordinate all the different types of activities, from kind of low levels of activity, small adjustments of your limbs, all the way up to jumping and running. Okay, these are defined as either slow or fast, and there's two types of fast fiber. And our bodies, if I melted us down, are approximately 50% fast and 50% slow. Okay. And the reason for me going into this will become apparent in a second. So in dystrophic muscles, the fast muscle fibers are more susceptible to damage due to the lack of dystrophin. This has been known for over 30 years. Back in the day when small muscle samples were taken from kids with Duchenne, and they saw that the fast fibers were breaking down more often than the slow fibers. We've designed an investigational therapy, Edge 5506, as Abby mentioned, to protect those fast fibers from damage. Slow muscle fibers are not directly affected. So what we're really hoping to do is nip the disease in the bud in the most susceptible muscle fibers. In animal models of muscular dystrophy, Edge 5506 protects fast muscle fibers from injury, improves overall strength, and limits muscle wasting and the long-term development of fibrosis, including an unexpected finding for us, which is cardiomyopathy. So we have an impact both on skeletal muscle and cardiac muscle in these early models of muscular dystrophy. We believe that protecting fast fibers may prevent further muscle degeneration and slow disease progression. A phase 1 clinical trial to test the effect of 5506 is ongoing. Let me give you a little more detail on what 5506 does. Now, I mentioned that myosin is involved in muscle contraction, and muscle contraction is the event that kicks off all of the muscle degeneration that you saw in that video. 5506 selectively inhibits that protein in fast fibers only. So it's a selective inhibitor of fast myosin. So our goal is to limit myosin to a level that prevents breakdown but not healthy contraction. By using some clever tricks, we've engineered a small molecule, 5506, that selectively modulates just half of your muscle, leaves the other half intact. By dosing to the right level, you can protect the muscle, but leave it functional. In animal models of muscular dystrophy, Edge 5506 resulted in decreased CK and other biomarkers, as well as increased activity. And I will show you two snippets of data from the preclinical package. So we're taking this kind of structural approach. We're trying to relieve stress in the skeletal muscles, targeting these susceptible fast fibers. As you can imagine, we're not altering the dystrophin status. So in the context of Duchenne, for instance, we're not changing the fact that the muscle doesn't have Duchenne. We're just altering the skeletal muscle so that it can deal with not having dystrophin. Okay, so as such, we're really designed to stop the damage where it begins. We're going to nip it in the bud. We're going to try and stop the muscle fibers from breaking down, but it doesn't preclude us from dealing with other therapies, things that restore or replace the dystrophin. So the microdystrophin AAVs that are in development, or the antisense oligos that are already provisionally approved, and then of course downstream type of approaches such as glucocorticoids. We're right at the top. We should really have a disease modifying effect, but we're very interested in how well the compound works in combination. We would expect it to be additive at the very least. Now, you saw a video of muscle breaking down. This is an example of 5506 at work in dystrophic muscles, same kind of video system. So these are dystrophic muscles from a mouse. Again, they're very small. We're looking at them through a microscope, and we're contracting the muscle in order to push it over the edge, get this excessive stress to cause breakdown. The left-hand side, we haven't really added anything. This is just the muscle contracting just in buffer. And on the right-hand side, we've added a small amount of 5506 just to modulate how much internal stress is present in that muscle. So about now on the left-hand side, what you can see is exactly the same thing as you saw in the original video. You start seeing these breakage events where the muscle is being snipped up, and then you can see the fibers retracting, and then more and more of these kind of hypercontracted blobs occurring, which are really just the whole length of the muscle that's crushed down, ready to be disposed of so that the body can grow a new fiber. And you can see these accumulating more and more on the left-hand side. Now on the right-hand side, still a dystrophic muscle, still no dystrophin, but we've normalized the internal stress, and now the thing's just contracting as if it was a normal muscle. There's no hypercontractions, there's no calcium entry, there's no membrane stress. The muscle is essentially functioning as if it was a normal muscle, albeit without dystrophin. I'll show you one more piece of data, and this is from the most complicated model that we ran in the dystrophic dogs. So, of course, if we could arrest that muscle breakdown in your whole body, targeting these fast fibers, then there's a couple of things that you might expect to happen short term. One would be the circulating biomarkers of injury, things like creatine kinase, would be reduced because the muscle's no longer stressed and there's less protein leaking out of that injured muscle. And the other thing that you might observe is that if you could restore those muscles to a more healthy contractile state, then your strength and your function would improve. And we asked both of these questions in these dystrophic dogs. So what we did in a couple of different studies is took older dystrophic dogs that were symptomatic. So these weren't young puppies, these were older dogs that are already progressed in their disease and were weak, okay? Akin to an older Duchenne kid. First thing that we did is we gave the dogs a short-term dosing and looked at creatine kinase, and you can see pretty much after the first dose, we saw decreases in creatine kinase. That's on the left-hand side. The interesting thing is in a second study, we also saw increased activity. So we actually fitted these dogs with collars that had activity monitors on them, so you could just measure how active or inactive those animals were. Of course, we could have decreased creatine kinase by making those dogs less mobile, right? The less active they are, the less CK they're likely to reduce. But we saw the opposite. These dogs are actually more active when they were dosed with the compound, and their CK was lower, suggesting that you had altered the relationship between the muscles contracting and the muscles becoming injured. Now, if we could achieve this in individuals Becker and Duchenne, that would be a really good way to set off a long-term clinical study just to see if you can improve long-term impacts of the disease. At this point, I'm going to hand it over to Joanne, and she'll give you an idea of where we are clinically.
Thanks, Alan. So now we have, after all that work, now figuring out how does Edge 5506 work in people? Is it safe? How do we best develop this for patients with Becker and Duchenne? So we start, as always in drug development, we start with baby steps in phase 1. And we start with basically dosing the drug for a day at a time and then for a couple of weeks at a time and going up gradually in doses. So, and then looking at not just the safety, but also how much of the drug is absorbed, where it goes, and how adults respond to the drug. The interesting part, or the different part of this phase 1 study, is that we are also including participants that have Becker muscular dystrophy. And we have been enrolling these patients for a while. And it really, they deserve a gold star for stepping up with this, because this is as hard, and we know it's hard to be in clinical trials in general, but they actually stay at the phase 1 unit for 2 weeks. So they're staying in there. So for us, or for the people who are running the study, to be able to follow them closely, to take blood at different intervals, and to basically assess how they're reacting to Edge 5506. So we learn about the safety, we learn about how humans that don't have Becker and those that do have Becker, how they process Edge 5506. Is that any different? And importantly, Alan mentioned the biomarkers of muscle damage. We'll be able to look at those and see, because we know that those change in Becker and in Duchenne muscular dystrophy. So, and of course, as many clinical trials do, we have an independent data safety monitoring board of experts to follow the safety and make sure that things are going well before we go on to next steps. So that's all going on. One of the things that we are mentioning on the next slide is we are looking at biomarkers. And Alan mentioned that the fast muscle fibers, the ones that are targeted with Edge 5506, are actually affected early in Becker and Duchenne. Now, you've all heard of and seen the results on your sons for creatine kinase. And we know these scales on these charts are actually going up in factors of 10. So the levels of creatine kinase are very high in Duchenne. They're low in control adults and intermediate in adults with Becker. Now, one of the things that has been developed is an assay that specifically looks at a molecule, a protein that comes from the fast muscle fibers, fast troponin. You've heard about troponins that are measured for cardiac reasons. This is skeletal muscle troponin, and it's just from the fast muscle fibers. And interestingly, the patients with Duchenne have very high levels. The patients with Becker have intermediate levels compared to unaffected people, so that have very low levels of fast troponin. Now, the other half of muscles is slow troponin. That is that is not affected early on in Duchenne. And interestingly, you're not actually seeing the troponin very different from those muscle fibers. So the damage in Duchenne and Becker seems to be focused on the fast muscle fibers. And we'll be able to look at these in the phase 1, but also in additional studies down the line. Those are the kind of biomarkers that can tell us how the drug is working. Is it having an effect on the target, on what we're trying to accomplish in terms of affecting the myosin molecules in those cells? So in the next slide kind of shows where we're going. We are continuing with the phase 1 study, the healthy volunteers, as well as the participants with Becker. And we'll be sharing those data later in the year. Those are still ongoing, and we are looking forward to having all the data and being able to share it with you. In the first half of next year, we anticipate starting a phase 2 study in Becker. And that will be a longer study, and it will look at functional, not only biomarkers, but functional measures over the longer term. And after that, in the second half of next year, we'll be able to start a trial in Duchenne as well. And we haven't finalized the entry criteria or the design of that study, but you'll be hearing more about that. And basically everything that we are doing now in terms of the phase 1 is building a foundation, not just for Becker, but also for Duchenne, because we think that EDGE 5506 can be helpful in all forms of dystrophinopathy, and in fact, even beyond that, to other muscle diseases. So on the next slide, just to kind of give you an overview, I hadn't mentioned, but this is an oral drug. It has a long half-life, so it can be given once a day by mouth. And it would be potentially appropriate for patients of any age and any mutation. So as Alan said, it's not specifically targeting a mutation. It's targeting the process that leads to disease progression. So when the boys are young, their muscles are functioning pretty well. And what we want to do is interrupt that process by which the disease progresses. When we presented our data, our preclinical data, and the mechanism of action to FDA for fast track designation, they granted us fast track for Becker muscular dystrophy. That is, we believe that's actually the first time that has been granted for Becker muscular dystrophy alone. As I told you, we have the phase 1 studies that are enrolling adults with Becker for this 2-week study to measure biomarkers and safety. And we have a plan for the phase 2 study in adults and adolescents, because again, we see this in all ages. And we know that there are a number of adolescents with Becker. It's typically diagnosed at a younger age, and there aren't clinical trials for adolescents with Becker. So we are planning that, as well as an open label extension for that. We'll follow that phase 2 study, we anticipate. And we are looking to how do we develop this best for Duchenne muscular dystrophy with all of the evolving knowledge that is progressing in the field, to be able to design a study and to listen to you guys about how to best do a trial with the least burden possible, but to get where we want to be, which is to be able to bring a drug forward to change the lives and improve the lives of those affected by dystrophinopathies. We also think that EDGE 5506 can be used in limb-girdle muscular dystrophy, the same idea of protecting the muscle fibers, as well as other muscle diseases. So more to come on that in the future. And on the next slide, with that, as Abby promised, we have an email address where you can reach us. We are a small company in Boulder, but really all over the country, with the mission of creating life-saving medicines for muscular dystrophy and bringing them to the community with urgency. So with that, I will stop, and we will be happy to listen to your questions and answer anything that we can. Thank you.
Thank you. Thank you, Joanne, Abby, Alan. That was a great presentation. We really appreciate you bringing this presentation to Jet Foundation and sharing it with our families that we serve through our programs. And as we mentioned, if anybody listening here today has any questions, feel free to write them right in your screen there on the questions box. Also a note that this webinar will be recorded. So if you were logging in late or you missed something, it will be recorded and on our website in our archive library within the next day. So yes, please go ahead and send us some questions. I see some coming in now. Okay, the first question. What age range and ambulation status will you target in your DMD study?
Great question. I think that we haven't finalized it yet. What just in general though, our sense is that if we, based on the mechanism of action, the more we can protect muscle early, and that's of course the case you're seeing with a number of clinical trials, the more you can protect muscle early, the more effect you can have. And the idea in clinical trials is to, and what the FDA has shared in its guidance, is that it's okay to kind of look at a group where you can see an effect faster, because ultimately that advances clinical development faster overall. Because they will take it that if you show an effect in one group, and if that's younger boys, then they will potentially then say, yes, we understand, we want to know it's safe, but we then can reasonably conclude that it's effective in a larger group of patients of different ages. So where we are also looking at patients with Becker as well as with Duchenne, we will be then looking at patients over a broad range of ages, and that hopefully will give us the information to be able to guide over a broad age. So I didn't give you an age range at this point. We haven't nailed that down, but that's kind of where we're thinking in terms of probably the younger boys. So who can do functional tests that we would be looking at.
Great, thank you. The question here is, could you explain how EDG 5506 strengthens muscle strands?
That sounds like a me question. So, you know, we've used cripes. We've used so many different types of analogies. Is a complicated concept, right? You know, I often use the concept of a truck. So imagine you have a vehicle like an old truck, and that thing's rolling along, but every time it hits 60 miles an hour, the wheels fall off. Now, of course, it still looks like a truck, but that is not going anywhere anymore. You have to repair that truck. You have to put the wheels back on. So we essentially have an approach in 5506 where you put a speed limiter on that truck that limits you to 59 miles an hour. Now, you're not going to break any speed limits, but what you're going to do is have a truck with wheels that's going along. And you can use that analogy in many ways for a dystrophic muscle, where normal functionality can be approached by just limiting how much that muscle can injure itself. So that's how you can strengthen muscle. You know, you can imagine it by looking at those videos. If a proportion of your muscle is breaking down like that every time you use those muscles, it's going to limit how well they function, because the body's too busy dealing with all the inflammation and the breakdown. If you can nip that in the bud, if you can prevent that breakdown, then you have a functional muscle that can still do its things. You know, it can still metabolize, it can still produce force, and it can inspire health, right? So that's our goal, is to really restore those muscles to a normal range of functionality.
Great. Let's see, next question is, how will you be confirming a Becker diagnosis? Strictly in-frame deletion or functional diagnosis?
So what we've heard from the experts in the field, we know that the vast majority of patients who have Becker have a in-frame diagnosis or in-frame mutation, so that they are producing dystrophin, but it may break down quickly. It may have, it may not be functionally what it's supposed to be doing. So that, and therefore, when they used to do biopsies more, they would see that there was some dystrophin there. So patients with an in-frame diagnosis, with a typical clinical picture would be the patients. But we also know that there are patients that have an out-of-frame mutation that have Becker. So it has to not just be a genetic, it has to be a combination of genetic and clinical diagnosis. So that's what we are thinking about. That's what we've heard from the experts in terms of making that diagnosis and trying to include. There used to be kind of an intermediate group, and I think that people are using that term less, that either Duchenne or Becker.
Great, thank you. Next question, how does the maximum force of MDX mouse muscle treated with EDG 5506 compare to that of a healthy mouse muscle?
Oh, that's a very good question. Cripes. So, of course, force is something that we artificially measure by electrically stimulating a muscle via the nerve. Okay. The target inhibition level for 5506 at the top end is about 20%. So, for instance, in that video that you saw of 5506 protecting the muscle, we can get that effect by decreasing the force in the muscle by 15%, 1.5. So that muscle is contracting at 85% of normal. If you do something technical like a grip strength test in an MDX mouse, that level of inhibition is not enough to measure a difference. So you actually, in a functional context, when you're measuring strength, you don't see any deficit at all. It's happening. It's just that our clever bodies are working ways to get around that level of a block. So we're really hoping just to take the maximal amount of stress on the muscle, and that appears to be enough to restore the muscle to normal functionality.
Great, thank you. Okay, our next question, have you studied older mice to see if EDG 5506 has regenerative benefit?
Ooh, well, I'm going deep here. Yes, we have done everything. I've tortured my pharmacology group for the last 4 years, frankly, asking every single possible question. And what the longest term study that we've done, and this is actually in our regulatory filings if you really want to go there, but the longest term dosing studies that we've done are 17 months in an MDX mouse. Now, it wasn't a regular MDX mouse, it was the more severe DBA2J MDX mouse, so pretty much the entire lifetime of a mouse. And at that age, you don't really need to do detailed biology. You can just see the difference. The mice look really terrible at that age, and the compound really has a profound effect on spinal curvature and on atrophy of the muscles. Because of course, with age in mice, much the same as in older Duchenne and Becker individuals, the regenerative power of the muscle goes away and you get muscle atrophy. The muscles actually waste away a little as well. So we do have a pretty nice effect long term in models.
Great, thank you. Okay, 2 more questions here. Are you actively pursuing combination strategies with exon skipping and gene replacement therapies? It's kind of a 2-part question. And number 2, is it okay? Is CK an established predictive surrogate endpoint for FDA and EMA? You need to break that down.
Yeah, one for Alan, one for me.
Yeah, I'll start with the science bit and then you can go into the practical bit. So obviously there's a number of questions that you can have for combinations, and we are interested in all of those. Now, my first question is glucocorticoids, because everyone is on a background of that if you have Duchenne. Not so much Becker, but we want to know, does the compound work well with deflazacort and prednisolone, for instance? Is it additive? A number of people have suggested that the steroids functionally improve things because they stabilize the membrane. Now, we do that too, but from inside the muscle, and our hypothesis is that we will be additive with those approaches. We certainly would be doing clinical trials in the early stage in the background of those agents. So we want to know that. Are we additive with steroids? So we're doing preclinical work in that area. In terms of the ASOs, well, there's a couple of questions that you can answer. Does the mechanism still work in the background of an ASO? I think there's every reason to believe that. There's also a secondary interesting question. If we stabilize the muscle, does the efficiency of an ASO get better, right? So if you use Exondys in the background of 5506, do you see more dystrophin? So those are questions that we're really interested in. And in many ways, the same questions are appropriate for AAV microdystrophins. If we stabilize the muscle before you give an AAV microdystrophin approach, do you see more gene expression, right? So we're asking efficiency of those two approaches and efficacy of our approach in the background of those therapies. And it's our desire is really to help build a landscape where people have options, right? This is a very severe disorder, and we're hoping to really add something significant into the armament of therapeutics to make a big difference. Joanne, I will pass it over for the clinical bit.
Okay. And I think that we have all learned that trying to predict what FDA is thinking is not a great exercise, and it's hard to do. I think what they are looking for is they are looking for to see everything hang together. So we know that CK goes around, up and down in patients, but what it doesn't do is go down and stay down. So, you know, if you could see a difference in CK that was sustained, that would be meaningful, and it wouldn't be the only thing that they would be looking for. But I want to be very clear that that is not the case. But what it is, it's part of the whole package that they are looking for. And to us, it's also kind of trying to understand proof of concept. And so it was very reassuring to see it in the models of dystrophinopathy and seeing the CK go down. So that's how we're looking at it. It's part, I mean, we have to look at many different aspects of the disease in order to be able to say definitively and to convince the FDA.
Great, thank you. Did you try EDG 5506 in aged mice that were not treated early in life?
Well, I guess I haven't really done that specific question in a mouse, but we did do it in a dog, right? Which is physiologically more relevant because dogs, their muscles are very similar to ours. So most people doing studies in dystrophic dogs start their study when the dog doesn't have any symptoms. So when they're very young, they're kind of like a normal pup. And then they'll put them on the medicine, and then they'll look 6 months later at how that disease has progressed. We didn't do that. We allowed the disease to progress so that those muscles were in a pretty bad way, and the dogs were weak by the time we gave them the compound, and we still saw the CK and the functional modifications. So I think that's a reasonably good proof of concept that in a disease established individual, so where some fibrosis has kicked in, that you would see an improvement.
Great, thanks, Alan. This says one last follow-up question on old mice. Would you consider an aged mice study combining EDG 5506 to stabilize muscle with GCSF to mobilize stem cells?
Goodness gracious, that is very specific. There's a few things that are not in your favor with that experiment. And one of them is that the old mice with muscular dystrophy aren't that bad. So if you take a regular MDX mouse, that's the standard mouse model of muscular dystrophy, even an old mouse is fairly good. and their stem cells aren't bad. So they're not like an old patient. Now, you could do that study in the DB82J, but they've been specifically engineered that their stem cells don't work. So mobilizing their stem cells would be very, very difficult. So I would argue that you could try that type of approach, but the mouse may not be the best place to do it. Now, in terms of combining, for instance, with an agent that might increase stem cell mobilization, you have to imagine that an approach like 5506 prevents the muscle from breaking down in the first place. So you don't actually need the stem cells, right? Stem cells are required when you have breakdown and you need to repair. If we can stop the muscle breaking down in the first place, you don't need the stems to repair anything. And that's a really profound differentiation right there. That's why we say we stop the disease where it starts. We stop the breakdown completely. We can stop the need for stem cells to mobilize to repair your muscles. So that's a real goal for us.
Great, thank you, Alan. More of a comment, it looks like here, knowing that you are studying in dogs, the EDG 5506 and GCSF study would be better in dogs.
Yeah, it would. It would. That would be the place to do it. Yep. Dogs, of course, very challenging model, but that would be a more disease relevant place.
Is next question, is your goal to stop or delay progression?
Stop. That's the goal right there. Don't want the disease, we want it to stop. Now, of course, the studies will have to show that, but that's the goal.
And what are you thinking of using for outcome measures?
So I think that they are pretty well described, certainly in the ambulatory, the younger boys, in terms of things like the North Star and the time function tests. So those are clearly in. I mean, that's what everybody is actually using very similar measures at this point. I think that what we haven't decided on is kind of looking at other potential measures. We're certainly going to look at patient parent reported outcomes as well, so we haven't decided yet. We're a year away from the Duchenne study, so we haven't decided yet if there are any other additional measures that we'll be incorporating in the study. So more to come on that.
Great, thank you, Joanne. Okay, I don't see any more questions. So if anyone has some more questions, please feel free to add them to the question box. But I would like to say, Abby, Joanne, Alan, this was a great presentation. You guys did a great job answering the questions. I think the questions coming from the community are showing that people are really excited. And so we're really grateful for you guys to bring this webinar to us and take the time to answer so many questions. And a reminder again that if anybody missed part of this webinar, it is being recorded and it'll be up on our website in the next day, that you'll be able to rewatch and share at any time. And with that, I don't see any further questions. So Edgewise Therapeutics, thank you all so much for spending this time with us. And we look forward to hearing more great things from all of you. So
thank you for everything you do.
Thanks.
All right. Take care, everyone. Thanks so much. Have a great afternoon. Bye-bye.
Bye.
Bye.